cosmetics that, according to manufacturers' claims, guarantee success with the opposite sex can be purchased not only in specialized adult stores, but also at any pharmacy or perfume shop. But how much trust can we really place in the promises printed on the labels of these Casanova perfumes? The influence of pheromones on human sexual behavior has only recently begun to be studied. Information that reaches the general public is often based on speculation, guesswork, and research funded by companies that produce trendy love elixirs. Pheromones in insects and certain mammals have been studied far more thoroughly, so let's begin, as we should, with butterflies. As early as the 19th century, French entomologist Jean-Henri Fabre suggested that male butterflies locate females by smell. This was confirmed only in the late 1950s by a group of German scientists led by Adolf Butenandt. In the name of science, they used 500,000 female silkworm moths to extract all volatile substances from their sex glands, separate them using chromatography, and demonstrate that only one of these substances caused male moths to flutter their wings. The compound was named bombicol, after the genus Bombyx, to which silkworms belong. Soon similar substances were discovered in many insect species, as well as in amphibians, reptiles, fish, and mammals. The role of pheromones in mammals has been studied most thoroughly in the context of sexual behavior and reproduction. For example, experiments on mice have shown that females prefer the scent of dominant males. That same scent suppresses sexual maturation and activity in other males, reducing their chances of reproducing. Moreover, the odor of a dominant male can cause early termination of pregnancy, allowing the female to conceive again and potentially produce more viable offspring. After numerous experiments with laboratory mice and rats, scientists concluded that animals relying primarily on their sense of smell in intimate matters tend to choose mates whose major histocompatibility complex MHC genes differ as much as possible from their own. The proteins encoded by these genes not only trigger tissue rejection in transplants but also play a crucial role in immune system efficiency. The greater the diversity of these genes, the broader the range of antibodies the immune system can produce to fight infectious agents. Ultimately, this diversity determines an organism's survival and its ability to adapt to changing environmental conditions. Naturally, researchers hypothesized that a similar relationship might exist in humans. One method used to test this hypothesis is the so-called sweaty t-shirt experiment, in which women are asked to smell worn t-shirts from different men and choose the most pleasant scent. In most cases, women prefer men whose MHC genes differ maximally from their own provided they belong to the same race. This may be explained by the fact that excessively large genetic differences could result in non-viable offspring. Possibly for this reason, many white individuals find the natural body odor of black individuals unpleasant, while some Asians perceive the scent of Europeans as sharp and sour. Research on larger animals has yielded even more striking results. It was found that androstenone, a metabolite of the male sex hormone testosterone present in the urine and saliva of boars, induces a mating posture in female pigs, even when no boar is nearby. Androstenone has also been detected in human male saliva and sweat. Meanwhile, vaginal secretions in women contain copulins, whose analogs in monkeys reportedly drive male rhesus macaques into a frenzy. These compounds became the basis for the production of sex perfumes intended to attract the opposite sex. And fortunately for lovelorn hopefuls and aspiring Casanovas, experiments have shown that airborne androstenone does not increase women's sexual desire. Some data suggest that it may improve women's mood and make them feel calmer and more confident, but nothing more. As for copulins, no measurable effect on men has been reliably demonstrated. Unlike insects or tadpoles, mammals do not exhibit such pronounced instinctive responses to pheromones, and these responses involve higher nervous system activity. A moth glued to a glass slide will flap its wings in response to the scent of a female of its species no matter what, whereas a well-trained dog can be deterred from joining a canine mating frenzy with in humans, with our relatively underdeveloped sense of smell and highly developed brains, detecting the influence of odors on behavior is extremely difficult. It is not even clear whether we possess a specialized organ dedicated to pheromone detection. Ordinary odors are perceived by animals and humans alike through the olfactory epithelium in the upper part of the nasal. Because pheromones play such a crucial role in animal life, reptiles and most mammals possess a specialized structure for detecting them, the vomeronasal, or Jacobson's, organ. The long projections of its neurons lead not to the main olfactory bulb but to an accessory olfactory bulb, and signals about pheromones reach higher centers of the central nervous system in parallel with information about regular smells. For a long time, it was believed that the rudiment of the vomeronasal organ that appears in human embryos atrophies during prenatal development. In fact, this organ is also present in adults. It was first described in the early 18th century by a Dutch physician who operated on a soldier with a facial wound, but this discovery was undeservedly forgotten for a long time. More than a century later, scientists described the vomeronasal organ in various animal species, and only relatively recently was its existence in humans confirmed. In humans, the vomeronasal organ appears as a small pit located about 15 to 20 millimeters from the edge of the nostrils. Approximately 80% of people have these pits in both nostrils, the rest have them in only one. However, no accessory olfactory bulb, specialized nerve fibers from the vomeronasal organ, or even a clear pathway for transmitting nerve impulses from its epithelial cells has been identified in humans. Nevertheless, increasing evidence suggests that volatile substances released by other members of the species Homo sapiens, even when not consciously perceived as odors, play a noticeable role in our lives. In 1998, American physiologist Martha McClintock demonstrated that applying underarm secretions from one woman to the upper lip of another synchronized their menstrual cycles. This finding explained her earlier observation that women living together often experience synchronized cycles. Further research showed that male scents also influence women's menstrual cycles and timing of ovulation, and that the smell of sweat from sexually aroused men activates brain regions in women associated with sexual behavior and pleasure. Thus, despite the lack of definitive scientific proof of human pheromones, attributing these and other long-standing observations to their effects seems logical. For example, girls who grow up in the same household as their biological fathers tend to experience puberty later than girls raised without fathers. A slight delay in puberty has also been observed in girls attending all-female schools. Today, it is generally accepted that human pheromones likely do exist and consist of fragments of sex hormone molecules and possibly other substances secreted by sweat glands and modified by skin-dwelling bacteria. The uniqueness of each person's microflora ensures the individuality of their scent profile, including a pheromonal component that is virtually odorless in the everyday sense. This may explain why a nearly blind infant recognizes its mother, and why adults can easily identify clothing worn by close relatives or loved ones. Modern civilization offers effective tools in the fight against pheromones, sanitary products, deodorant, antiperspirants, and antibacterial soap. Perhaps these products partly contribute to the romantic difficulties many people experience today. However, abandoning hygiene is not a solution. The smell of an unwashed body would easily overpower any subtle enhancement of pheromone. And fortunately, or perhaps fortunately, the idea of a universal pheromone capable of attracting the opposite sex is untenable. Any perceived effectiveness of sex perfumes can most likely be attributed to the suggestibility of consumers and the boost in confidence they experience. Even if such a universal pheromone were discovered, it would likely be useful only for finding a one-night partner.